Welcome to this explainer. Today we're diving into the ultimate biological factory inside your very own cells. We're going on a trip that takes us from microscopic assembly lines operating at just blinding speeds all the way to the fundamental science of how and why we age. It's this beautiful, complex process that literally keeps you alive, but ironically enough, it's also the exact same process ticking away your biological clock.
500. Just think about that number for a second. That's the staggering amount of nucleotides, the basic building blocks of DNA, that a simple bacteria can add to a new DNA strand every single second. 500 per second. It completely blows my mind and brilliantly illustrates the sheer speed of this biological process. Just imagine a factory churning out 500 complex components every second with near-perfect accuracy. That's happening inside living cells right now.
So, to make sense of all this, we're going to hit four main stages today: unzipping the double helix, the DNA replication machinery, prokaryotes versus eukaryotes, and finally, telomeres and cellular aging.
All right, let's jump right into part one, unzipping the double helix.
Stepping onto the factory floor, our master blueprint is the famous double helix. But here's the catch: it is anti-parallel. Think of it literally like a two-way street. One strand of DNA runs in what biologists call the 5' to 3' direction, while the opposite strand runs in reverse, from 3' to 5'. This traffic pattern, it's going to pose a massive engineering challenge for our factory workers in just a minute. But before we even get there, we actually have to pry these two strands apart.
Now, you can't just rip DNA apart anywhere, right? It all kicks off at a very specific spot called the origin of replication. And get this starting line is absolutely packed with adenine and thymine, or AT base pairs. Why? Well, because A and T are held together by fewer hydrogen bonds than the other pairs. They're literally the path of least resistance. It makes the DNA strands so much easier to separate so the factory can finally start production.
Moving right along to section 2, the DNA replication machinery.
You can't run a factory without a specialized crew, and in your cells, these are your essential enzymes. First up is helicase, which acts like this little engine driving forward to unzip the DNA. But DNA desperately wants to snap back together, so single-strand binding proteins act like tiny wedges to hold it open. Then primase lays down a tiny starting block, an RNA primer. Once that's all set up, the real star of the show, DNA polymerase, starts building the new strand, nucleotide by nucleotide. And finally, ligase comes in like glue to seal up any breaks.
So, putting it all together, the assembly line sequence makes total logical sense. You unwind the raw material, drop a primer so the machinery knows exactly where to start, meticulously build out that complementary strand, and seal the gaps. It's a continuous, beautifully flowing process, well, at least on one side of the street.
Because remember that two-way street we talked about earlier? Yeah, our star builder DNA polymerase can only work in one direction, 5' to 3'. This means while the leading strand is built smoothly, the opposite side, the lagging strand, actually has to be built backwards in these small disjointed pieces. These pieces are called Okazaki fragments, named after the brilliant Japanese scientist who discovered them. It's just a spectacular biological workaround, letting the factory build backwards while the whole operation keeps moving forwards.
Okay, let's look at section 3, prokaryotes versus eukaryotes.
It is super fascinating to see the massive contrast in scale between different organisms handling this exact same job. Prokaryotes, like simple bacteria, they're basically a scrappy single-origin startup. They replicate their simple circular DNA right out in the open cytoplasm from just one starting point. Eukaryotes, on the other hand, like mammals and, well, us, function way more like a massive multinational corporation. We replicate complex, linear DNA safely locked inside a nucleus, using over 50,000 different replication sites simultaneously just to get the massive job done.
And you can really see this difference in their production speed. Because the bacteria's machinery is simpler, it just blasts through replication, adding 500 to 2,000 nucleotides per second. Meanwhile, our mammalian cells take a much slower, highly managed approach, adding only about 50 to 100 nucleotides per second. Basically, we trade raw speed for meticulous, careful management of our massive genomes.
Actually, even the physical size of those backwards-built Okazaki fragments is totally different. The simpler prokaryotic machinery churns out these massive, chunky fragments of up to 2,000 nucleotides. But our highly regulated eukaryotic machinery produces much smaller, finer pieces, only about 100 to 200 nucleotides long. The core process is the exact same, but the execution perfectly reflects how complex the organism is. Finally, we arrive at Section 4, telomeres and cellular aging.
So what happens over time? Well, this biological factory equipment inevitably begins to suffer from microscopic wear and tear. And this brings us right to the science of aging. Because eukaryotic DNA, our DNA, is linear rather than circular like a bacteria's, a massive engineering problem pops up. Imagine painting a floor, right? But you paint yourself into a corner. How does the replication machinery copy the very tips of the linear DNA strand without just falling off and losing crucial genetic data?
Biology's workaround for this is absolutely brilliant. It uses telomeres. Telomeres are special repeating sequences of nucleotides at the very ends of our chromosomes. Think of them exactly like those little plastic caps on the ends of your shoelaces. They don't actually contain vital genetic code for building you. Instead, their entire job is to sacrifice themselves, taking the structural hit so the valuable genetic instructions inside aren't destroyed during the copying process.
Here's the crucial takeaway, though. This factory equipment naturally degrades. Every single time your cells divide and DNA is replicated, the machinery just can't copy the very end of the line, meaning the telomere gets slightly shorter. Biologists call this the end replication problem. Over a lifetime, these protective caps physically wear down, and this gradual shortening is directly linked to cellular aging and the hard limits of the human lifespan.
But wait, it gets crazier. There is actually a specialized enzyme called telomerase that is capable of rebuilding these tips, essentially extending the telomeres back out. This enzyme is highly active in our stem cells and germ cells, which obviously need to divide countless times. But, and it's a big but, unfortunately, telomerase is also dangerously hijacked by many cancer cells. This allows them to solve the end replication problem for themselves and achieve uncontrolled, theoretically infinite growth.
This whole dynamic of shortening telomeres is the absolute foundation of the chronographic theory of aging, originally proposed by Alexei Olovnikov. His theory suggests our lifespans aren't just wearing out randomly, they're strictly dictated by an internal molecular clock. The shortening of those telomeres acts as a literal chronograph, a timer counting down the replicative limits of our cells.
Which leaves us with this absolutely massive, provocative question to wrap up this explainer. If cellular aging is, at its core, simply a mechanical issue, just a shortening of a physical sequence, and if we could fully decode forced telomerization without accidentally triggering cancer, could we eventually pause the factory's degradation? Could we one day permanently hack the human biological clock? It's a question at the very frontier of modern biology, and it all traces back to how your cells copy a simple strand of DNA. Thanks for exploring this with me.